Okay, then let's talk about FDRs and about protein inference. I mean, I know that some of you already moved into that going through the tutorials. I apologize for that, but it's very hard to sort of have the talks exactly at the point when everyone needs them, and you all move at very different speeds. So occasionally we have to explain a few things after you already stumbled upon it in the tutorial. What we are doing with is really trying to understand how reliable the identification is, and we're now talking about the PSM, the peptide spectrum match, and I will get back to that later on. It's essential to know what FDR we're really talking about, because there are different FDRs. FDR is a very general concept, and it's meaningless if you don't specify what you're trying to estimate FDR. When we're looking at databases, we would of course expect that the database contains all the potential candidates for our search. It is not so obvious to figure out how you would make sure that you can also get incorrect identifications, because if you want to make statistics on the identifications, it is good to know which of them are correct identifications and which are definitely incorrect. And it was a pretty smart idea when people started using Target-Decoy databases, because that gives us a very intuitive understanding of this false discovery rate. The idea is fundamentally very simple. We take the original database, let's say it contains 20,000 sequences, and those we call target sequences, because that is what we're really trying to hit in a database. Now, what you can do is you can add 20,000 sequences of which you're sure that they shouldn't occur in your sample. And there's a very simple way to do that. You can just reverse them. I'll show you what that looks like in a minute. And the idea would be you do two searches. You search against the target database, you search against the decoy database. And all the hits that you get from the decoy database have to be wrong. These sequences should not occur in nature. But it gives you a score distribution of incorrect identifications. And you can use that to compare that to the score distribution of the correct hits. You can imagine it like this. So you run a search, and you can actually do these two searches in one. You search against one unified database. And let's just say these here are the peptides that come up, and we search them by the score. We rank them by the score. So this here is the best score, and these scores get gradually worse. Some of these hits will come from the target database. Some of these hits, however, will come from the decoy database. Now, you would expect that the hits against the target database are on average better than the hits that you get from the decoy database, because they will just be random matches, and the target hits should be correct matches of the whole spectrum.
Is this the same parent mass, or we're talking about different parent masses, different fragments from S1 to
We're
S2?
talking different PSMs, yeah? Different target masses, different spectra. Because these guys here could not have the same parent mass. So this is the whole data set that we're now looking at. Now what you can do is you can decide, I accept everything up to a score cut of, say, S6. And of course this list is not just 15 peptides the way I showed it. In reality, this would be 100,000 hits that you have. And then you can go over that list once you've ranked it and decide everything above this score I trust, and everything below that score I do not trust. So how do you decide where to make this cutoff? Well, that is where the FDR comes in. Let's look at how these scores come about. We talked about computing these scores earlier. When you look at these score distributions, you can usually fit something like a mixture model to them. Yeah? So if this is a score and this is a frequency, then these hits will come either from the decoy database or from the target database. So on average, these decoy hits will be worse, so they probably have a distribution like this. I'm not saying the distribution should exactly look like this, but for simplicity's sake, let's assume that's what they look like. And then you have a second distribution. These are the correct hits. Now, for some scores, it becomes hard to keep them apart because they're perhaps crappy spectra, have a low score. But some of them have a very high score, and here it's evident this is going to be a correct hit. Now, what you can do once you have modeled that, you can determine what is the probability, given such a score, that this was really a right hit. These target decoy databases simplify now the computation of such a false discovery rate. Once you have determined that ranking, you can basically check how many target or decoy hits would you get if you cut off at a certain point. These decoy hits you would discard anyhow, you know they're decoy hits, but they basically tell you how likely it is that what you see is incorrect. Why is that? Because once you get into this gray area down here where you cannot keep targets and decoys apart, you will get decoy and target hits with equal probability. If you are in the upper part of that list, you will have way more target hits than decoy hits. So you can basically say, if I cut here, well, what is my false discovery rate? Well, we can simply count. If we cut up here, we have 1, 2, 3, 4, 5 targets, 6 targets, 7 targets, and 1 decoy. So it's 1 over 8. That is my false discovery rate at this point. And of course, if you have a longer list, you get better statistics out of that. But that is the very simple idea. And you can compute this FDR for every point of this list, and then you can make up your mind. How many of these false discoveries do you want to have? Sometimes it's better to have a very low false discovery rate because you really want to trust every single point and you have enough data. Then you say, I accept only 1% FDR. But sometimes you want to be more sensitive. Since you're going to validate everything anyhow, you can say, I want to discover as much as possible. So you accept 10% FDR. And you know, there's not a single correct FDR. You have to choose the FDR in a way that is acceptable to your problem. Because if you put the FDR too low, you lose many identifications. If you put it too high, you get too much incorrect peptide hits. And you need to find the right balance for it. So the second thing I want to talk about is the protein inference problem. So far, we have not identified a single protein, so we haven't done proteomics. All we have done is peptidomics, if you want. So we really need to go one step further, going from peptide identification or PSM identification to protein identification. So on the PSM level, we can now compute this FDR. And again, that does not, if you have 1% FDR at the PSM level, that does not mean that on the protein level you have the same FDR. We will see what that looks like. It can be vastly different. When we look at these identifications, then you would assume it's straightforward to go from the peptide sequence to the protein sequence. You could blast your peptide sequence, for example, against the protein database, or you can look where it occurs, which peptides it came from, or proteins it came from. Now we need to talk a bit about this uniqueness. If we're interested in proteomics, we really don't want to quantify peptides. We really want to quantify proteins. That means we need to map it back. And the uniqueness, unfortunately, depends on many parameters. It depends on the size of the database. If you have a database that has lots of redundance, then you have multiple hits for the same peptide in there. It also becomes more likely for longer peptide sequences, that is not surprising. The expected value that they hit the same sequence in the database just goes down with the length of the sequence. What are the reasons for this uniqueness? I mean, one of them is chance hits. You can imagine if you're looking for a peptide of length 2, what are the odds of finding that guy anywhere in a sequence, where in a random sequence it's 1 over 400, 1 over 20 times 20 for an arbitrary sequence. But we also have other biological reasons, and one of the reasons is that peptide protein sequences are not random. We have gene duplication going on, we have homology between sequences that we see, and of course we have protein isoforms that are basically sharing many of these peptides. And if you want to keep those apart, you need to dig a bit deeper. So how does it look like with the uniqueness? This is a simple comparison of different databases, in particular RefSeq and Ensemble and Tremble, that have different degrees of what happened to my mouse pointer? There we go. That have different degrees of redundancy. And what you can see is that while the total number of triplet peptides goes up, actually the number of unique peptides levels off. So at some point you are starting to get the same sequences over and over again. You can also see that the uniqueness depends on the molecular weight or on the sequence length. This here is for various databases, the degeneracy. So if this approach is 1, then the peptide is unique in the database. Again, we see a similar behavior that's not unexpected. As you go to smaller peptides, you see the same sequence over and over again. And if you go to larger databases, you have some redundancy in the database as well. One of the issues here are splice variants and isoforms of proteins. And there's a figure here that illustrates that to some degree. What do we really mean by uniqueness? If we have peptides that come from different proteins, there are different ways how this peptide can be unique. So let's look at these different guys here. So we got 4 genes here, and illustrated here with these boxes are the exons of these genes. When you look at, and then this here is a eukaryotic proteome. So if you have a peptide that occurs only once and only in one isoform, in one exon, then if you identify this guy, then you're sure that you have found one specific isoform, isoform 1 of gene 1, and nowhere else in the proteome will this sequence occur. So if you found this peptide and you're sure about it, then you're also sure that your sample contains isoform 1 of protein 1. It's different for this exon here. Same gene, 1 exon in front of that. If you find this peptide, then it can come from this exon here, but it also occurs in another gene. So all you know if you see this peptide is that you have either gene 1 or gene 2 or both of them, and you can't keep the 2 apart no matter what you do. And in this way you can sort of arrange these identifications in various degrees of uniqueness. Some of them are unambiguous. This is unambiguous on the isoform level. This guy here is unambiguous on the protein level. But others are ambiguous, so if you identify one of them, you have only an idea that several of these guys need to be present, at least one of them, but you don't know which one. So how often does that occur? Is that something that happens routinely that you need to deal with? Is that something that is rare? I gave you the numbers earlier. When you go into Nextprot and you search, that's probably the best resource we have right now for human protein isoforms at least. There are on average 2.96 different splice variants per protein. So splice variants do happen. Some of these proteins have vastly larger families. So this here, PDE9A, you can look that up in Export if you like, that has 16 documented isoforms. And you see while large parts of the proteins are shared between isoforms, up here in front there are many things going on that make it really hard to distinguish these 16 isoforms. And basically, if you identify something here, then you have no idea which of these isoforms it is. And if you find something down here, then it depends really on which isoform it belongs to, whether you can make any statement about that at all. So what that gives rise to is this notion of protein families. The problem really is we should not imagine that we see the whole sequence covered. In reality, something like this here is more frequent. Highlighted here in black are all the peptides that were actually observed in a sample that contains this gene. That is what we call protein coverage. You should not imagine that you really see a peptide for each of these subsequences. So why not? What happens to the other peptides? Why is this guy not there, this guy?
Some of them don't ionize well.
Yeah. What other reasons?
Some of them are too hydrophilic, stick to a column.
Yeah.
Sharp.
Or some are too hydrophobic and just stick to the tube. Others have been degraded by proteases at the terminus. So there are many different reasons why you don't see them. It's also a matter of concentration. You can imagine some of them have a very low copy number. And then you run into effects that it becomes hard to really detect that. So we will, in very rare cases, will you see more than 50% of these proteins covered. For the most part, it is individual peptides here or there, so you see only a fraction. If we want to resolve that, it depends a bit on what type of proteome you're dealing with. In prokaryotic proteomes, over 90% of the identified peptides tend to be unique. That has to do with the genome structure and the complexity of the genome structure. In eukaryotes, if you have a large number of orthologs and if you have many splice variants, the number of unique peptides can easily drop below 50%. And that makes it hard, because if they're non-unique, are they useful? And the answer is yes, of course. If you deal properly with them, they are still useful, they still convey information, even though you might not be able to quantify everything exactly based on one of these peptides. So in literature, people have been talking about protein families, and protein families are ultimately different proteins that share peptides, and that where confusion might arise. These here are a bunch of proteins, and you see that one of them contains all the peptides, 11 peptides that are observed, and others contain only parts of that. And that does not necessarily have to be protein isoforms. It can also be shared sequence, can be reuse of domains. There are many biological mechanisms that lead to shared peptides. There are different ways to deal with that type of ambiguous information. And Occam's razor, or maximum parsimony, is sort of the most restrictive way of doing it. You probably know of Occam's razor. The idea here would be to find the smallest set of proteins explaining all observed peptides. So what would that mean in our particular case? Well, if we observe, for example, 4 peptides, and 3 of them are also contained in another protein, then the easiest explanation is just taking this protein and not making any statements about B, because whatever we say, we cannot really make a statement about B. So we just reduce a number, we just discard B. B could still be in the sample, but we would say the best explanation of what we see is A. Why is A the best explanation, not B? Well, the first peptide here, this guy, that cannot be explained by B. So if we assume A is present, then we explain all 4 of them, and that is better than explaining just 3 of them. So there are different types of ambiguity here as well. Distinct proteins do not share any peptides. Yeah? So you cannot confuse 2 distinct proteins. Differentiable proteins can be distinguished by at least that 1 distinct peptide. So either that peptide is there or not. That turns them into something that you can distinguish or something that you can potentially confuse. The indistinguishable proteins, like this guy here, no matter what you do, will not be able to keep A and B apart. You will only be able to make statements about A and B, never about A or B. And there are subset proteins, like this guy here, and there are subsumable proteins that contain peptides that are also contained in other proteins. Yeah? This, for example. In this case here, if you have A and C, then that would explain every observation of B that we have. Usually this is reported as so-called protein ambiguity groups, PAGs. And there's an example here. We got, again, 4 peptides. Protein A contains all 4 of them, B contains 1 through 3, and C contains 2 through 4. In this case, if you observe 4 peptides, what would you report? Well, with a parsimonious approach, you would just report A, because that would already explain everything. But this would not mean that B and C are absent in the example. So if you really want to be sure what you report, you would report this whole protein ambiguity group and say, I see 4 peptides, and they come from this family of proteins, ABC. And then you need to use different approaches to figure out what is really going on. Can you think of other ideas on how you can disentangle such a protein ambiguity group?
Antibody, you can pull all 3 down theoretically.
Yeah, you could do it with other theoretical approaches. Why would the interactions have you?
So they are not independent proteins. So for some of the isoforms, you have more connections to the other identified proteins.
Yeah, but that's a very complicated approach. One of the things you could look at is quantitative data. Yeah, if you have not just 1 sample, but if you have 20 samples, and if the concentrations of these 3 proteins change in a meaningful manner, then you can look at the ratios of those guys. Why is that interesting? Because you should be able to observe that if this guy here changes independently of that guy, then it cannot be all A. Yeah? So it has to be, well, either B and C, or it has to be A and A, either of those. Yeah? But you can, through this type of argument, you can start inferring what is really present in the sample and what is not present. But from the observation of just the qualitative data alone, you will not get very far. So what you usually get out of these protein inference algorithms is something like this. You have a bunch of peptides and a bunch of proteins. And then they are reported like this, where you have protein A that is identified through peptides 1 and 2, protein B through peptides 3 and 4. And there you see now things can get a bit complicated if the same peptide maps to multiple proteins. Then you can see that multiple proteins are supported by the same peptide, like B and C through peptide 4. And you will get groups where we cannot really make out whether all of these are present. So with B and C, we are sure that they are present because peptides 3 and 5 are unique to them. But with this protein group, H, I, and J, here we have so much ambiguity that we cannot really say with certainty that either of these proteins is present. We just know that at least one of them has to be there. And if that is in your report, you need to know, you just need to decide whether you care about each of these proteins enough to try and figure out with other experiments whether they're there. Now, I would like to get back to the significance of these hits. What's the meaning of a PSM for protein identification? Well, ultimately, if we compute the FDR at the PSM level, then we'd have no conclusion on the PSM or on the FDR rate at the protein level. This is totally independent, needs to be determined independently. 1% FDR on the PSM level means that 1 out of 100 PSMs is probably incorrect. But that doesn't tell you anything about the proteins. Why not? Proteins have different lengths, have different peptides, and oftentimes you get the same PSM over and over again because the same spectrum is identified over and over again. It's also a function of abundance that you see there. If you have PSMs for a large number of mostly identical samples, then what happens is that the correctly identified hits, they are always there. But what happens to the incorrect identifications? Well, they accumulate because they're random. So if they're incorrect, these false discoveries sort of scatter over all the sequence space. So if you actually accumulate 100, 200 samples, you also accumulate false identifications. And that leads to a very different picture on the protein rate. So historically, people have talked a lot about how to deal with that problem. And there was a very pragmatic approach that was actually written down in Paris Guidelines for Data Deposition in Proteomics, where people said, discard these single-hit wonders. So if you have a protein that is supported by only one peptide, discard it. You should not report it. That is not a good idea. You remove data that you can still use, and it's a very ad hoc rule. Why not discard also all the proteins that are supported by only 2 peptides? Also, you see there's some dependency on the length. So this single-hit wonder thing, this 2 peptide rule has been discarded again. And there have been a couple of studies that give you some idea that what we observe is not totally straightforward. So why did people want to get rid of these single-hit wonders? Because they said we want to get rid of these false positive identifications that accumulate. And on the PSM level, that is also true. So here we have, that's taken from a paper where people looked at the number of peptides in the target database that were identified based on the single peptide rule and the 2 peptide rules. And you see you get more correct target hits at the same FDR. However, these are PSMs. They are not proteins. And curiously, if you do that on the protein level, the picture is exactly inverted. You actually get fewer protein identifications if you do it that way. The statistics behind that are not really straightforward. But having more peptide identification, that does not necessarily mean that you have more protein identifications. If you do the math, and you can do something similar that we did for Target-Decoy on the protein level as well, the statistics are not really straightforward, which is why we will not go into the detail. I just give you some idea of what happens. But this simple example here explains how you can get with a 20% error rate at the PSM level already to 40% FDR at the protein level. And oftentimes, I know that there are papers that had 1% FDR on the PSM level reported. And what do you think? How high can the protein FDR go if you have 1% PSM FDR? Well, if you accumulate enough false positives, they can get pretty high. So there's some papers where the protein FDR is about 50%. And that is why I said earlier that the FDR at the PSM level is not really what you might be interested in. Because if you accumulate enough data sets, throw them all together, then you get FDRs on the protein level that are not what you would like to see. And then you hand your collaborators a long protein list that you collated based on these PSMs, and 50% of them are just nonsense. And that's just not a good idea. All right, I'll stop here. Half an hour is over. I hope I could give you some idea of at least a rough outline of what FDR and protein inference means, and I'd be happy to take any questions, and otherwise we'll just continue with the handouts. Thank you.